Good morning to everyone. I am a principal researcher at CIMAR, which is a research center of the University of Porto, where I am the leader of the animal health and aquaculture team. The topic for today is nutritional immunology as a tool to boost fish and shrimp health condition. And I will start with a very brief introduction to both the immune defenses of fish and shrimp, and then I will jump to more practical, let's say, case scenarios where I will talk a little bit about how nutrition can improve, for instance, the health of these interesting animals. So fish and shrimp are in intimate contact with their environment, which can contain very high concentrations of pathogens, as we all know. Under normal conditions, fish and shrimp maintain healthy state by defending themselves against these potential invaders by a complex system of innate defense mechanisms. These mechanisms provide protection by preventing the attachment, invasion, or multiplication of microbes in tissues. Their importance is threefold. Firstly, the protection is non-specific and does not depend upon recognition of the distinct molecular structure of the invading species. Secondly, there is no or little time lag for them to act. Even during the inducible defenses like inflammation, which are relatively quick to respond, for instance, about 1 to 2 days. So pathogens will have little time to establish themselves. And thirdly, they are relatively temperature independent. For ectothermic vertebrates, these characteristics are very important because the specific immune defenses take time, considerable time, to respond and are very temperature dependent. So as you can see here in this part of the slide, there is just two clear separations of the innate branch of the immune response and the adaptive branch of the immune response in fish. As you will probably know, shrimp only relying on their innate immune response. So on the one hand, in fish, we have the innate immunity, which has not memory, reacts with equal potency with repeated exposure, while during the adaptive immunity, there are memory cells that remember specific pathogens and antigens, and they will respond more effectively on a second exposure. However, recent evidence in both plants and invertebrates indicates that phagocytes, myeloid cells, can respond much better to a challenge if they have been pre-stimulated with the same or with another agent or antigen. So it leads to the thought or to the theory that innate immunity can have a memory also different from acquired immune memory. Particularly, innate immune memory has also been observed in myeloid cells from vertebrate lineages such as fish or even mammals, and this has been called trained immunity. So indeed, compared to terrestrial animals, aquatic animals have greater challenge because they need to cope with high microbial loads, eventually can bombard them through their mucosal epithelial barriers. The main mucosa-associated lymphoid tissues, or also called MALT, in fish are the skin-associated lymphoid tissue, or SALT, the gut-associated lymphoid tissue, or GALT, and the gills-associated lymphoid tissue, or GALT, which particularly the GALT is unique for fish. Recently, it has been discovered also the nasopharynx-associated lymphoid tissue, which apparently there are a particular special subpopulation of the cells in that area, and this is leading to innovative solutions for spray vaccination to improve the immunization of these animals. In this slide, you can see an overall overview of the both innate immunity and adaptive immunity in fish, where you can see the physical defenses in pink linked with humoral defenses in some cases, in light blue, and in dark blue cell defenses. So you can see the different hematopoietic organs, such as the head kidney, where it is located, the thymus, the spleen. So basically, lymphocytes and myeloid cells are used by the head kidney and can maturate in the spleen, while in the thymus, T lymphocytes are the ones that are being matured. As there is no time to go into the detail of this, of all these aspects, and the picture cannot include everything. So there is here, as you can see, there is no information about myeloid cells such as macrophages, which play an important role in antibacterial defenses, because these are the primary cells involved together with neutrophils in the phagocytosis of bacteria, and they can kill them principally by the production of reactive oxygen species through the so-called respiratory bursts. So these are important cells that we need to consider during innate immunity. And for instance, also the neutrophils are rich in myeloperoxidase granules in their cytoplasm, and through the myeloperoxidase, in the presence of halide ions and hydrogen peroxide, they can kill bacteria by halogenation of the bacterial cell walls. So neutrophil and macrophages are also very important because they contain humoral factors such as lysozyme and other hydrolytic enzymes in their lysosomes. For instance, these macrophages can also produce nitric oxide, which is a very important and potent microbicidal agent. Here you can see some integumental innate defenses and humoral factors. Many observations suggested the importance of innate defenses in the integument of fish. For instance, in a practical case would be the comparison of the rainbow trout and the Atlantic salmon, which are both salmonid species, where we already know that the rainbow trout is more resistant to immersion challenge with Aeromonas salmonicida compared to Atlantic salmon. So this suggests that the integumental defenses of the rainbow trout regarding against this particular pathogen are more potent compared to Atlantic salmon. So if bacteria are successful by encroaching the integumental defenses, there are a number of plasma proteins which may prevent further spread of infection. So we have in the first place the inhibitors, which will inhibit or which should avoid the proliferation of the pathogen. For instance, transferrin will avoid the availability of iron to the bacteria. Interferon will avoid the virus replication. Antiproteases will also block, let's say, proteases from bacteria. So these are a number of mechanisms very important in diseases. The lysins are also very important proteins that will have an important role in destroying the pathogens, and we also have a group of precipitins and agglutinins, such as petraxins, for instance, which are acute phase proteins and very, very, very important during the inflammatory response, as well as the importance of lectins, such as proteins that will bind to carbohydrate, to peptidoglycans, to glycolipids and glycoproteins that are within the cell surface or at the surface of different pathogens. Some of these proteins mentioned are also present in the mucus, and will also and may also be in higher concentration, such as the case of lysozyme. Lysozyme is very abundant either in the serum or in the mucus. So it's actually important humoral defense at both systemic and mucosal levels. So in terms of the different listings in immunity, we have the proteases, which are known that present in the skin mucus of many fish species and contain trypsin-like activity. The lysozyme is, as already told, is a lytic enzyme which hydrolyzes N-acetylmuramic acid and N-acetylglucosamine, which are constituents of the peptidoglycan layer of bacterial cell walls. Lysozyme has been found beyond serum and mucus, has also been found in different tissues in fish. Antimicrobial peptides are very important also as innate immune defenses. They are small peptides whose actions have great implications in the innate immune response of teleost fish, being considered critical as the first line of defense against a wide spectrum of pathogens. Here I created a couple of them, the group of the piscidins and the defensins, because these are the antimicrobial peptides with wider, let's say, with a broader range of capacity to kill different pathogens. The complement system is a serum. It's a system of serum proteins, which is central to many defense mechanisms. The complement system is composed of more than 35 soluble plasma proteins that play key roles in innate and adaptive immunity. Complement is initiated by one or a combination of three pathways: the classical pathway, the lectin pathway, and the alternative pathway. The classical pathway is initiated by a complex between an antigen and antibody. The alternative pathway is initiated by the spontaneous activation of the complement component C3, which is amplified upon the covalent binding of C3 to various microbial surfaces such as viruses, bacteria, fungi, parasites. And last but not least, the lectin pathway requires the interaction of lectins such as mannose binding lectin or, for instance, ficolins with half sugar moieties found on the surface of microbes. Well, central to the adaptive response, we have the lymphocytes, which are cell types responsible for the diversity of antigen recognition, specificity, and memory. On the one hand, we have the B lymphocytes, which are responsible for antibody production, and on the other hand, we have the T lymphocytes, which are responsible for cell-mediated immunity. As you probably know, fish like bone marrow, where lymphocytes originate in mammals, but they have the head kidney, which you can say that it's analogous to the bone marrow. And here both B cells and T cells originate. However, the maturation of these cells occurs in the head kidney, whereas the maturation of T lymphocytes, it will happen in the thymus. T cell receptors and B cell receptors are extremely diverse molecules at their antigen binding type. These, for instance, in the case of B cell receptors, these are plasma membrane anchored immunoglobulins. In contrast, B cells are responsible for the secretion of immunoglobulins, which are also known as antibodies. In teleost fish, there are three classes or three types of immunoglobulin isotypes that have been identified as IgM, IgD, and IgT. T cells are subdivided into cytotoxicity T cells that directly kill infected and abnormal cells, the helper T cells that modulate other immune cells through cytokine production. And among the helper T cells, we also have regulatory subsets, also called Tregs, that are involved in the regulation or suppression of immune responses in order to prevent immune-mediated damage. In this slide, you can see an overview of the fish immune system, how it starts with innate immunity, usually at the physical barriers to prevent pathogen entering. Once the pathogen can enter into the host, there will be a series of cell-mediated immune defenses and humoral defenses, and through the cell defenses, innate immune cells, such as macrophages and dendritic cells, will present antigens to the T lymphocytes through the MHC glycoproteins that will interact with B lymphocytes to release antibodies. To contribute also to the humoral defenses. Also, after the recognition of the pathogens, there will be a process of inflammation that will eventually can activate the defense process, and of course, ideally would lead to the pathogen destruction. So, talking now about shrimp immunity, crustaceans have a hard outer cuticle that also acts as a skeleton in that muscles are attached to it. The cuticle covers the entire outer surfaces of the animal, including the gills, and the anterior and posterior regions of the gastrointestinal tract. Therefore, many disease-causing agents that need to gain entry into the underlying tissues must be able to penetrate through this challenging barrier. However, there is a shortcoming, which is the evolutionary design of this solid cuticle, which leaves crustaceans unable to grow. So they require periodic episodes of molting when a new cuticle is formed, so the old cuticle is set. The problem here is that the new cuticle is initially very soft, and this leaves the animal susceptible to both predation and disease. So it will also interfere in the immune status of the animal, and when you are analyzing the immune status of shrimp, you should also consider the phase where the molting cycle is beginning. So this is very important where evaluating health condition of shrimp. We also have the peritrophic membrane, which is known to play an important role in the defense system of arthropod gut, and particularly in the white leg shrimp seems to be a key issue, since the digestive tract is considered one of the major issues targeted. One of the major tissues targeted by the white spot syndrome virus. In shrimps, hemocytes are the main cells responsible for immune defense. Three main types of hemocytes have been identified: hyaline cells, which are granular cells, semigranular cells, and granular cells. Sometimes these cells are called semigranulocytes or granulocytes. However, the term granulocyte applied to crustacean hemocytes can be misleading and should be avoided. Okay, since there is no evolutionary link between these invertebrates and vertebrate leukocytes. So beyond that, like all invertebrates, shrimp do not have cells of the lymphoid series. There are no antigen-presenting cells, such as dendritic cells, and there are also no natural killer cells. Hyaline cells are amoeboid with granular cytoplasms, and they are the professional phagocytes in shrimp. So they are responsible. They are the main responsible for phagocytosis. On the other hand, semigranular cells and granular cells have limited phagocytosis function, but they are responsible for the encapsulation, cytotoxicity, and also for the storage and release of the peripheral oxidase system, which I will tell you about that and the importance of this system. The humoral defense includes in shrimp clotting, the melanization of damaged tissues, and also pathogens by the peripheral oxidase system, activation of antimicrobial peptides, and the release of estrus-responsive proteins and molecules believed to function in opsonization and iron sequestration. There is also a cellular response once the pumps from the pathogens are detected, and mainly alien cells will go through phagocytosis, so they will engulf small pathogens like, let's say, bacteria, and for instance, metazoan parasites or big, let's say, pathogens with a big size, they cannot be engulfed, so they will be encapsulated, and through the process of melanization, they will be destroyed. So, as I told you previously, the prophenol oxidase system is very important. It's of key importance in the defense of arthropods. The central enzyme is the phenol oxidase, which is produced when the inactive form peripheral oxidase is released from the cytoplasm of granular cells as a result of stimulation. There are a variety of pathogen-associated molecular patterns, also called PAMPs, that can activate the stimuli, including, for instance, peptidoglycan from bacteria, LPS from bacteria, beta-glucans from fungi, among other possibilities. The products of this enzymatic cascade, the peripheral oxidase system, include melanin and a variety of potentially toxic kinones. The clotting system is also very important in the humoral response. It can be considered also as the first line of defense and an integral part of the overall invertebrate immune system. It is also important in the prevention of blood loss during injury and wound healing. And also we have other very important humoral factors, similarly to that found in fish, such as, for instance, antimicrobial peptides, lysozyme, and lectins. So jumping now to more practical case scenarios, let's say, as you all know, the aquaculture intensification is increasing more and more, and this will bring stressful events to the animals. For instance, in the case of fish, the stressor will be perceived by the brain, which will release a series of neuroendocrine and even endocrine responses through the release of cortisol and catecholamines into the bloodstream, which will lead to secondary stress responses, among when we find the metabolic responses to cope with the stress situation, immune responses to cope with the stress situation that are transitory. So these secondary stress responses will allow the animal to reach homeostasis again, or in the case of the stress event persists, there will be tertiary responses that will be at the whole organism level. So we will see behavior alterations at the behavior level. We'll see decreasing growth and unsuccessful reproduction, and also higher susceptibility, because there will be an immunosuppression, and mainly due to the chronically of risk of cortisol. So when this happens, many farmers or many strategies usually go to the use of antibiotics, which is not sustainable and will lead to the to antibiotic resistance. So there is the need to go through other prophylactic strategies, and nutrition is one of these possibilities through the functional feed application. So as you probably know, through functional feeds, we can increase the pigmentation of the muscle, increase the pigmentation of the skin, improve gut health, improve growth. And among all these applications, we also have the enhancement of the immune system through different supplements such as probiotics, prebiotics, minerals, vitamins, pigments, and amino acids. During the last years, the team I'm leading, we have been working with methionine and indispensable amino acids, and particularly in the case of amino acid methionine, we observed that you can improve the immune status of the European sea bass through the increase of peripheral leukocyte responses, increase of complement activity, and bactericidal activity. We also observed that there is an increase in the inflammatory response through the improvement of cellular responses, humoral responses, and cellular recruitment to the inflammatory focus. So in a recent study, we decided to try to get some more details into these mechanisms, and European sea bass were fed either a control diet or 2 diets supplemented with methionine. This was DL methionine to 0.5 or 1% of feed weight. And after 2 weeks of feeding, animals were infected with our threat pathogen. We observed that there was an increase in the peripheral leukocyte response, and also there was an increase in the peritoneal. Sorry about this. It says peripheral here, but I was supposed to write peritoneal. So peritoneal cell populations, because with these, the cells we are collecting here at the peritoneal cavity, we can see which type of cells are being recruited to the inflammatory focus. So we injected the bacterial pathogen into the peritoneal cavity, and after 24 and 48 hours, we collected those different cell types, and we observed that there was in fact an improvement in the total leukocytes, neutrophils, and macrophages at the peritoneal cavity. And we also observed a time effect. So this means that all animals fed either experimental diet were able to improve the different leukocyte types, either at the blood level or at the peritoneal cavity level. So we also observed that after this bacterial challenge, the animals fed with those diets supplemented with methionine showed an improved disease resistance compared to those fed the control diet. We also can improve the diets, or can develop functional diets for fish using algae. Algae are very important ingredients because they have bioactive compounds. Bioactive compounds such as highly unsaturated fatty acids, cell wall polysaccharides, and also pigments and polyphenols. For instance, HUFA can improve the pro-inflammatory responses because they are precursors of prostaglandins, or they can also, through other mechanisms, improve the anti-inflammatory response and be immunomodulatory. Through other components of algae, such as cell wall polysaccharides, we can also observe some improvement in gut wall protection at the level of the gastrointestinal tract, and also some immunostimulatory properties. And also through the pigments and polyphenols, we can see some improvement in the oxidative stress response. So in a recent experiment, we fed head cerebrine during 1 week or 2 weeks with different diets supplemented with the inclusion of 3 different microalgae and 2 different seaweeds, and we observed changes in the level of the blood and at the level of humoral components. And interestingly, we did not observe much changes after the 1 week or 2 weeks of feeding, but after stimulation with the bacteria, we observed a clear increase in the neutrophils population, and mainly after 1 week of feeding, we observed a huge increase in the neutrophils population in those animals fed with the ULBA diet. Microalgae can also be included in microdiets for Senegal salt porch larvae. In a recent study, we observed that there was an increase in the final dry weight in Senegal salt porch larvae fed with the Nanochloropsis diet compared to the control diet. And the other, also not clearly significant, for instance, in the case of Phaeodactylum, we concluded, or we suggested, that this improvement in final dry weight could be related to the lower total glutathione content in porch larvae, which could be linked to the higher antioxidant capacity of these different microalgae through the pigments that are present in this interesting. And in a recent study, it was also observed that extracts from 2 different seaweeds can improve the health condition of the white leg shrimp. And as you can see in this graph, there was an improvement of the survival of shrimp after submitted to a challenge with Vivipara hemolyticus. Therefore, the seaweeds can also be applied to shrimp nutrition, to shrimp health, and this could be a very interesting tool to improve, to apply as prophylactic strategy eventually in shrimp farming. And so, to sum up, I would just like to show you some most frequently evaluated immunological parameters when we are using diets to improve the health condition of animals. In the case of shrimp, we usually see different counts at the level of the red blood cells, white blood cells. In the case of shrimp, counts on hemocytes also, sometimes just barely total hemocytes, but ideally it would be nice to see the differentiation between granular and granular cells, and also usually the respiratory parts through the ROS production is also measured. And at the level of humoral components, we have different innate parameters such as proteases, antiproteases, peroxidase, lysozyme complement, and lymphocyte in the case of fish, and also usually the IgM is also measured in serum or plasma. And at the level of shrimp, we usually see the peripheral oxidase system, such as one of the most important and more measured parameters. Also, lysozyme proteases, antiproteases, and nitric oxide. So, just a few words before ending. I would like to tell you that it is very important to keep in mind that immune responses are very species specific. We cannot apply a particular diet to an animal if we see that this diet is improving the health condition of another animal. So we need to know very well the species which we are working with, and we need to have reliable protocols and tailor-made specific diets for those animals. Age and breeding conditions are very important. It's not the same to work in ponds or in RAS system. It's not the same to work with post-larvae or with juveniles. And availability of additives and dosage is very important. We need to know how much we can add, how much you should include in the diets. There is a risk of toxicity. There is a risk of no change at all. So we also need to know about this, the importance of this, of the type of additives and the amount of additives. Last but not least, the feeding regime is also important. If we are aiming to immunostimulate, this should be short feeding regime. We cannot immunostimulate for long-term periods because this will be detrimental to the animal. So keep in mind that immunostimulation is a prophylactic strategy and not a long-lasting. And I would like to thank you for attention.